Blessed day, everyone. This is Richard devera again. Now, our topic is all about the transcription and translation. This topic is also one of the important that why our body is amazing. Because if we talk about transcription, this is where the process of gene copying. And translation is all about producing protein in our body. That is important, especially we are exposed to many organisms that should not be needed in our body. So now we will start to know how our body is amazing and how God is amazing. First, we have to know about some description of transcription. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. This is the process of copying out the DNA sequence of the gene in the similar alphabet of RNA. Oh, amazing. The main enzymes involved in transcription is the RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the 5 to 3 direction, adding each new nucleotide to the 3 end of the strand. We have the steps of transcription. The first step is the initiation. It is the beginning of transcription. Of course, it occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. The second step is the elongation. This is the addition of nucleotides to the mRNA strand. The RNA polymerase reads the unwound DNA strands and builds the mRNA molecule using complementary base pairs. There is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During this process, an adenine and the DNA binds to an uracil in the RNA. And the third step is the termination. This is the ending of transcription and occurs when RNA polymerase crosses a stop sequence in the gene. The mRNA strand is complete and it detaches from DNA. And we have also the stages of transcription. Transcription of gene takes place in 3 stages: the initiation, the elongation, and termination also. Here, we will briefly see how these steps happen in bacteria. You can learn more about the details of each stage. The first stage is the initiation. This is where the RNA polymerase binds to a sequence of DNA called the promoter, found near the beginning of a gene. Each gene has its own promoter. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template needed for transcription. The promoter region comes before the transcribe region, whose transcription it specifies. It contains recognition sites for RNA polymerase, or its helper proteins to bind to. The DNA opens up in the promoter region so that RNA polymerase can begin transcription. And the second stage is the elongation. When one strand of DNA, the template strand, acts as a template for RNA polymerase. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotides, making a chain that goes from 5 to 3. The RNA transcript carries the same information as the non-template strand of DNA, but it contains the base uracil instead of thymine. RNA polymerase synthesizes an RNA transcript complementary to the DNA template strand in the 5 to 3 direction. It moves forward along the template strand in the 3 to 5 direction, opening the DNA double helix as it goes. And the third stage of transcription is the termination. This is called the terminator signals that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. An example of termination mechanism involving formation of a hairpin in the RNA. The terminator DNA encodes a region of RNA that forms a hairpin structure, followed by a string of U-nucleotides. The hairpin structure in the transcript causes the RNA polymerase to stall. The U-nucleotides that come after the hairpin form weak bonds with the A nucleotides of the DNA template, allowing the transcript to separate from the template and ending transcription. So that is the transcription. We are now going to translations. If we look our body, of course, this is made of skin, bone, and muscle. And each of those cells contain many millions of proteins. And our body is made up of cells. As a matter of fact, proteins are key molecular building blocks for every organism on Earth. So, I said earlier that translation is important because they are responsible for producing protein. Because we are exposed of our environment that so many organisms that enter in our body, we need these proteins to make us strong, and our body can depends itself. So there are steps of translation. Your cells are making new proteins every second of the day, and each of those proteins must contain the right set of amino acids linked together in just the right order. That may sound like a challenging task, but luckily your cells are up to the job. Amazing. There's also steps in translation. The first step is the initiation. The ribosome assembles around the mRNA to be read in the first tRNA, carrying the amino acid methionine, and which matches the start codon AUG. This setup, called the initiation complex, is needed in order for translation to get started. In order for translation to start, we have a few ingredients here: the ribosomes, which comes in two pieces, large and small, an mRNA with instructions for the protein will build, and the initiator, carrying the first amino acid in the protein, which is almost always methionine. Step 2, the elongation, is the stage where the amino acids chain gets longer. In elongation, the mRNA is read one codon at a time, and the amino acid matching each codon is added to a growing protein chain. And the third step is the termination, is the stage in which the finished polypeptide chain is released. It begins when a stop codon, UAG, UAE, or UGA, enters the ribosome, triggering a series of events that separate the chain from each tRNA and allow it to drift out of the ribosome. After termination, the polypeptide may still need to fold into the right 3D shape, undergo processing, get shaped to the right place in a cell, or combine with other polypeptides before it can do its job as a functional protein. And that's it, everyone. That is transcription and translation. I hope you learned from our topic. Have a good day and God bless.